Going to move ahead and discuss the cervical cancer. So first, of course, we start with the risk factors, which you study in a lot more detail in gynecology. So I'm not going to overlap that, but definitely I'm going to correlate it and tell you the meaning of some very, very important terms out here. When we say cervical cancer, all of you know that cervix is going to have squamous epithelium also. So human papilloma virus, of course, is one of the factors. Other than that, smoking, immunodeficiency, oral contraceptive pills, OCPs also have, and also multiple sexual partners. All of these are associated with cervical cancer. That is not all the things that I want to get into. What right now I want to ask you is that before cervical cancer occurs, what is the pre-malignant lesion that is there? And that is known as CIN. What is CIN? It is cervical intraepithelial neoplasia. It is cervical intraepithelial neoplasia. So now tell me one thing, till the time I'm saying something is intraepithelial means it is a precancerous stage. It is a precancerous stage, and they say CIN is of 3 types. Precancerous stage is going to be of 3 types, CIN 1, 2 and 3, and that is what I want to explain to you over here. So please remember, this is the normal cervical epithelium. This is the normal cervix that we have. I want to explain to you what is CIN 1, what is CIN 2 and what is CIN 3. So what is 1, 2 and 3? First we will understand those, right? Then I will teach you what is CIS and what is carcinoma. After this you will never ever have any concern understanding these terms. So what is CIN? Till the time I will use these terms called in situ or intraepithelial, in situ, till the time all these means, till the time I don't use the word cancer, make it a rule. I'll draw a division over here. Till the time I don't use the word cancer, make it a habit that your basement membrane is intact. Means all throughout over here, all throughout the rule is that no matter what happens, till the time cancer doesn't happen, everywhere I will just say that the basement membrane is going to be intact. Right? Now let's understand. What do I mean by CIN 1? So when I say 1, remember 1 for it. Where are all the bad cells seen? These green color cells are going to be bad cells, okay? Where are all the bad cells seen? Only in the lower 1/3. Means in CIN 1, 1 for 1, the lower 1/3 is going to show dysplasia. The lower 1/3 shows you presence of dysplasia. Then let's go to CIN 2. In CIN 2, it is 2 for 2. How much of dysplasia do I see? I see 2/3. I see now the bad cells are seen in lower 2/3. So repeating, what do you see in CIN 2? 2 for 2. This means the lower 2/3 is going to show you dysplasia. Coming to CIN 3. What is CIN 3? 3/3. What is 3/3? Means it's the full thickness almost. If you notice, almost, see, one cell is left, rest almost full thickness is affected. So can I say in a case of CIN 3, almost full thickness is going to show you dysplasia. There's almost full thickness dysplasia. Whereas when do I say full thickness dysplasia? That becomes carcinoma in situ. Carcinoma in situ means the full epithelium is gone. Starting from the top right till the bottom. There is CIS. I mean by CIS, carcinoma in situ means there is going to be a full thickness involvement. There is full thickness. Now get going with all the terms, guys. Repeat with me once again. CIN 1, 1 for 1 means the lower 1/3 has a problem. CIN 2, 2 for 2 means the lower 2/3 has a problem. CIN 3, 3 for almost 3. So almost full thickness has a problem. But when it is CIS, carcinoma in situ, means then proper full thickness has a problem. And after that, you can see what has happened. Now you have seen the basement membrane has broken. We know for a fact that as soon as the basement membrane will be broken, we have entered into a proper carcinoma. Are all the terms clear? But now the next problem that comes is, Many students say that now we are not following all this CIN1 and all of that. Now it has been replaced. What has it been replaced by? It has been replaced by something called LCIL and HCIL, right? So now I'm going to show you the same thing, but look at this. This was CIN1, this is CIN2, this is CIN3 and CIS, right? They say that this low grade CIN1 is known as LCIL, and all the others we can put them under HCIL. So what is LCIL and HCIL? Now they're making life a little easier. CIN1 is LCIL, means low grade. It is a low grade lesion. So L for low grade, squamous intraepithelial lesion. Low grade squamous intraepithelial lesion. Whereas what is HCIL? HCIL is high grade squamous intraepithelial lesion. High grade squamous intraepithelial lesion. So they made life a little simpler. They said only CIN1 is the low grade one, and all the others that you're talking about will become high grade. That will go into HCIL. That is the thing that is being followed nowadays. But more than this, in pathology, what I'm interested in is the screening modality that we use. What is the screening modality? It's a Pap smear. How do we screen for cervical cancer? We screen with the help of Pap smear. And now I'm going to teach you not only, a lot of gynae actually, not only am I going to teach you the procedure part, I will also be focusing equally on the infections, and then I will show you the cancer. So we quickly have to do a 20-25 minute discussion on that. So when I say Pap smear, please remember there are two techniques of doing a Pap smear. And before that, why do I call it Pap? Because the name of the scientist was George Papina-Colau. The name of the scientist is George Papina-Colau. That is the reason I call it Pap smear, Pap stain, Pap slide. Everything Pap is named after George Papina-Colau. There are two techniques. There is something known as a conventional technique of doing a Pap smear. So whenever I want to do something like a conventional technique, I am going to use what? I am going to use this Ayer's spatula. What do we use? We use the very, very famous Ayer's spatula. That's how you spell it. It's a very famous spotter that you get in your gynae practicals also. So that is the Ayer's spatula. But then they said, okay, what will you do? You will take the sample. After taking the sample, what shall you be doing? You will put it onto a slide like this. You will put it on a slide like this. It's like as if you're painting. So you took the sample and you put it on a slide. How do you take the sample? Let me show you. So have a look at this, guys. This is what you do. Whatever spatula, brush, whatever you'll have, with the help of a speculum, you are going to expose the area and you're going to take the sample from the cervix. So what is the lining of the cervix and what can I expect? I hope you all remember, cervix is composed of ectocervix and endocervix. These are the two things that you have to know, ectocervix and endocervix, and I think we know the basics of it. Ectocervix is made up of a squamous epithelium, and endocervix is made up of a columnar or a glandular epithelium. So this is simple, that when I'll be talking about endocervix, the cells that I will show you in pictures will be columnar cells. They are going to be long cells, right? The columnar cells are going to be long cells. Endocervix is going to look like this. But when I'll show you ectocervix, means I'll show you this particular part of the slide, I can see that there are many layers. 1 layer, 2 layer, 3 layer, many layers are there. So let me draw it. What are those many layers? Remember, PIS. What is PIS? So first let's draw. There are some cells which are going to be round at the bottom. These are known as parabasal cells. These are referred to as parabasal cells. After that, we can see that there are some cells in the middle. These middle cells are known as intermediate cells. These are in the middle, right? So they are going to be intermediate cells. And then you have some cells right at the top. So because they are at the top, I call them superficial cells. These are all the cells that you have in the ectocervix. So squamous epithelium, PIS. And it's so simple to learn. You don't need a mnemonic. At the base, you will have basal cells. So at the base, you have the basal cells. At the top, you will have the superficial cells. And in between, you will have the intermediate cells. As simple: base, superficial, and intermediate. These are the three cells that you will find in the squamous epithelium, the ectocervix. And what is the layer that you will find on the endocervix? You will find that tall columnar. So this is the sample. You are taking the sample from here. After taking the sample from here, what are you going to do? You are going to paint it on a slide like this. So now, don't you see two problems? When you were collecting the sample, did you only take the cells, or whatever must have been the blood in this area? Maybe the female was menstruating, maybe she has some infection, there's a discharge, there is blood, there is mucus, all debris, all dirty material. Have you collected that also? Problem number one. Problem number two, now when the pathologist will sit down to see this slide, pathologist will start viewing from here till here, corner to corner. It's going to be so time consuming because it's just painted like that in such a random manner. So it's so time consuming. Then they said it's better that we come out with something known as liquid based cytology. That is what they decided. So what was the purpose? The first question which you write as LBC, why did they come up with liquid based cytology? First question is, or the first reason is that it eliminates all the, it's done by a machine. I'll show you a machine. Liquid based cytology is going to be very useful because it will cut out all the debris. The blood will be gone, the mucus will be gone, all the dirty things will be gone. So it is going to eliminate all the blood and the mucus. It is going to eliminate all the blood and the mucus. Second, I'll show you a slide. The slide, the machine makes a slide which is just a round circle. It's not painted like this. So it is saving time. It is saving time of a pathologist. You have to just look at one tiny little area of 1 centimeter. You don't have to look at the entire slide. So it is saving time of the pathologist. But only saving time is not the concern. It is also definitely getting rid of all the blood, mucus. I won't have to look at unnecessary blood. I will see the actual infection, the actual cancer. I won't have to look at blood and mucus and all of that. That is the advantage of liquid-based cytology. So now let's get into the procedure part of it. So imagine for conventional, what were you using? For conventional, you were using an iron spatula like this. What were you doing? Taking the sample and you were painting it on a slide like this. Fine. On the other hand, when you are doing a liquid-based cytology, you are going to use a brush. Can you see this brush over here? You are going to use a brush. And why do I say liquid based? Because after taking the sample, you are going to put it in a liquid. You are going to put it in a liquid container. So obviously a question coming to your mind is, what is the liquid container having? It is having some kind of alcohol. So it could be ethanol, it could be methanol. Usually ethanol is what is preferred. So I'm going to take the sample. Now let me show you. What are they seeing? What are they doing over here? They took the sample from here. They took the sample. Let's see liquid-based cytology. They took the sample from here, and after this, where did they put the sample? They have put the sample into this container. Did you see? They have taken the sample and put it into this container because it has a liquid, it has alcohol. That is what is liquid-based cytology. But please remember now, liquid-based cytology is of two types. One is said to be sure path and one is said to be thin prep. So these are not some techniques. These are actually names of two machines. There is a sure path LBC and there is a thin prep LBC. And what's the difference? I'll tell you now. Look at this, guys. What have I done over here? I took the sample from the patient. So for example, if this is the container, this is my brush. I took the sample from the patient and in the brush, I have mixed it in the container very nicely. But I still feel some cells are left on it. I'm not very confident. So what will I do? I will break this brush. I will decide that I am going to break this brush and I'm going to drop the entire brush only into this container. I am going to drop the brush in the container. So remember, when you want to be double sure, when you drop the brush in the container, that is going to be a sure path. When you drop the brush in the container, there is no brush. This means there is thin prep. If you just rinse it and then you leave the brush aside, that is thin prep. But if I break the brush and I put it in the container, that is going to be sure path. So how I have learned it is, when you drop the brush in the container, you want to be very, very sure everything is going to the path lab. So wo pura brush only you send to the path lab, right? So remember, when you drop the brush in the container, that means you are wanting to be very, very sure that everything goes to the path lab. That is sure path. If there is no brush in the container, This means you are sending it for thin prep. So let's see, if I give you some diagrams, will you be able to identify? Look at this, guys. If they say this is a container and this is another container, and you don't have to know the process, but whatever the machine is doing, can you identify which is sure path and which is thin prep? Of course, because if I see the brush in the container, the gynecologist wanted to be very, very sure. So the brush was only sent. That is sure path. Here I don't see a brush in the container. There is no brush in the container. So this must be thin prep. Life becomes easy. So by looking at the brush, I can identify number 1. Number 2, finally, when a slide will be prepared. Finally, when a slide will be prepared, how will I differentiate whether it is sure path or thin prep? You will have to look at the diagram. Means look at this. The slide will have a circle like this. Again, I'll draw. The slide will have a circle like this. If it is a small circle of 13 mm. So remember that in sure path, number 1, you wanted to be very, very sure. So you decided I will drop the brush in the container. Sure for sure. Also sure for S for sure, S for small. The final circle that you get is very, very small. It is only 13 mm. On the other hand, if you are talking about thin prep, what is going to be the slide over there? You will see a bigger circle. You will see a bigger circle on the slide, which is going to be 20 mm. All this now that I have told you, have a look at it and equal slides. So first, can you see this slide over here, which is looking so dirty? This slide out here, guys, so dirty, as if you just took the iron spatula and painted it. What PAP is it? Conventional or LBC? This is the very, very time consuming one. This is the conventional PAP. Okay. On the other hand, then I see 2 beautiful slides. Circle over here and circle over here. So these 2 beautiful slides must be LBC. They must be the liquid-based cytology. Now, which is sure path and which is thin prep? This is the smaller circle which is 13 mm. This must be sure path. And then the other one is labeled also. You can see this is a bigger circle, almost 20 mm. So this must be thin prep. Will you be able to identify all of these? Yes, all's good. But did you notice something? I've done the technique. Going all the way back, I have done the technique. I know for a fact that yes, there is something called conventional, done with an iron spatula. We have something called liquid based, done with a brush and a liquid. We've seen the slides also. Conventional is going to be a dirty slide like this. Liquid is going to be a nice round slide like this. All that done. Why does this slide have a color? What stain have you used? So, what is the procedure of PAP staining? So, the first thing that you're going to do to this slide is fixation. After that, you are going to do a staining. These are the 2 things. Whenever a slide comes to you, for example, if this is a slide that has come to you, first you will add a fixative, then you will add a color. So, what do I do? What is the fixative? Previous year question, 95% ethanol. 95% ethanol is the fixative. Let me tell you, there are a lot of gynae books that mention it's a combination of ethanol and ether. Gynae books ke liye theek hai, but pathology books mention that ethanol is better. So 95% ethanol is a better fixative for pap smear. After that, you will put some colors. What colors should I be putting? So what is the mnemonic for pap staining? The mnemonic for pap staining is hope. What do I mean by that? So P over here is for pap stain. And hope basically stands for H for, see, pathology is pink and blue. Let's keep it pink and blue. So the blue color stain is hematoxylin. The blue color stain is hematoxylin. Then there has to be a pink color stain which is known as EA50. EA50 stands for eosin azure 50. Eosin azure. So eosin is basically the pink color. I've got a pink, I've got a blue. Other than that, they've also come up with something known as orange green. There is something known as OG6. OG6, so not the OG that you guys know on social media, but OG6, that is orange green. So there's a little bit of orange and there's a little bit of green also. Repeating, they could give you separately that which is what are the colors that it has. So it has a blue and a pink because everything in pathology is blue and pink. Other than that, is that it has OG6, that is orange and green colors also. So that's all that pap stain has. These are the 2 questions of your previous year. Question 1, what is the fixative of choice? 95% ethanol. What is the staining protocol? Hematoxylin, OG6, EA50. These are the pap stain processes. Finally, your pap stain is ready. This is your slide. It's looking colorful. It's time to look at it under the microscope. Under the microscope, what is the reporting methodology that we use? For reporting, we use the Bethesda system. We use the Bethesda system for reporting, and Bethesda system basically tells us not only for pap smear, remember, Bethesda system is not only used for cervix, it is also used for thyroid FNACs, it is also used for breast. I will be teaching you Bethesda. Bethesda is basically a place where a lot of scientists get together and they make all the rules and regulations which everyone in the country, all pathologists all over the world have to follow. So Bethesda is just a place. So when they decided that we'll sit at Bethesda and we are going to make guidelines for cervix, they came across with this. So what is the guideline? If you are a pathologist, say for example in Mumbai, I am a pathologist in Delhi, right? I say that I have a slide with me. I have seen this in Delhi. I think it is normal and everything is fine in it. When I send the same slide to you, you say, ma'am, there are hardly any cells in it. How did you say that it is fine? Is there a standardization between your reporting and my reporting? No, everyone will be reporting different things. So they decided we should first have an adequacy criteria, which is a previous year question also. So that if you are sitting in Mumbai and I am sitting in Delhi, you should also call that slide adequate and I should also call that same slide adequate. There should not be any discrepancy between my findings and your findings. So for that, we have to look at two cells. When I'm talking about cervix, what are the two cells? Number one, we have the ectocervical or the squamous cells, or the other one we have the endocervical cells. These are the two things. So now for conventional pap smear and for liquid based cytology, I realize the endocervical cell criteria is the same. Endocervical cell was what? The tall columnar cells. Minimum how many should be there? Minimum 10. Just 10 is the criteria. The same criteria is there for conventional pap smear also. The same is there for LBC also. So endocervical is simple. I should see at least 10 endocervical cells. The variation is in ectocervical cells. If we say conventional, then we need to see at least 8K to 12K cells per 10 high power field. And if we say LBC, 5,000 cells per 10 high power field. So repeating, 8,000 to 12,000 per 10 high power field for conventional and 5,000 per 10 high power field for liquid based cytology. So if I ask you, what is the adequacy criteria for conventional PAP? You'll say conventional PAP is 8K to 12K. I have to see ectocervical squamous cells and 10 I have to see endocervical. What is the adequacy for LBC? For LBC, you will say 5,000 I have to see ectocervical cells and 10 I have to see endocervical. Either ways, endocervical is always going to be 10 only. Everyone's understood that concept? Now you have done all of this, but do you remember I had only drawn these cells and shown you? Now it's time to look at the real cells. So what is the cells that I'm starting with? I'm starting with the same. First we'll go at the superficial layer, then we'll see the intermediate layer, then we'll see the parabasal layer, then we'll see the endocervix. Okay, in order. So let's start with the superficial layer. Superficial, PPP means superficial cells. P for polygonal. They are going to be P for polygonal. You can see the color, they are very pink. So yes, they are P for polygonal, they are P for very pink. And look at the nucleus, pycnotic nucleus. If I look at the nucleus, it's like a black color dot. It's like a black color dot, exactly. So we will see all the pink color cells, they are going to have a black color dot. Very tiny nucleus, pycnotic nucleus. So repeating once again, if they ask you how do you identify superficial cells, polygonal, pink and pycnotic. Superficial. Now let's come to what other color do I see in this picture? One is pink cells. Pink are all superficial. What is the other one? Yes, blue color cells. So the blue color cells are intermediate. How do I identify blue? They are blue and bigger. Big and blue cells are intermediate. So you can see definitely they are blue in color. But if I look at their nucleus, try to compare their nucleus is bigger. Rather than that pycnotic superficial wala, thoda sa nucleus is bigger. So remember, the big and the blue cells, the big and the bluer cells and the bigger nuclei, that is intermediate. Finally, coming to the third one. Before I teach you the third one, can you tell me what is this pinkish orangish cell? What do you want to call it? Superficial or intermediate? Superficial is pinkish, so this must be superficial. What do you want to call this blue cell with bigger nucleus, blue and bigger? This must be intermediate. What am I left with? When you'll see round cell clusters, these round cell, and they'll always be present in a cluster, these are referred to as para-basal cells. So now in the same slide, such a beautiful slide, I can see everything. When I go ahead and see these pink color cells, I am going to call them superficial, for pink, superficial cells. When I see the presence of these blue color cells are intermediate cells. And finally, when I see a cluster of these round cells, they are known as the parabasal cells. Is the entire ectocervix done? Superficial, intermediate and parabasal? Because now we can move on to the endocervix. So as we had discussed a while ago, endocervical cells are tall columnar cells. Can you all appreciate the same thing that we had discussed? Can you all see these tall columnar cells over here? They are as if there's a very famous name given to it, as if, you know, butterflies are sitting on a fence. This is said to be butterfly on fence appearance. This is said to be butterfly on fence, or also known as the picket fence appearance. This is the butterfly on fence or the picket fence appearance, because they are looking tall columnar. Again, now instead of showing you, so when I'm showing you the pencil like this, it is tall columnar. But if I say like a drone, look at it from the top. Now when you look at it from the top, it's not going to look like a long pencil. It's going to look like just one dot. So when I look at these cells from the top, just looking at the head of this pencil, if I just look at the head of this pencil, it's looking like a dot. So that is what it looks like from the top view. The top view is said to be the very famous honeycomb appearance. The top view is said to be honeycomb appearance. Let's do a repeat. When I see these endocervical cells like this, you're seeing it like this, it's looking like a tall columnar cell, butterfly on fence. I show it to you from the top view. From the top view, you're only seeing the round head, so it's going to show you the honeycomb appearance. Both of these are appearances of the same endocervical cells. How many endocervical cells do you need in the adequacy? Jaldi recall, try to put your mind into it. How many adequacy cells, adequate cells? 10 you need, whether it is conventional or it is LBC. Very good. Now, in this entire process, we forgot one thing. We have like somewhere become obsessed with only getting cells from the cervix. Are you only going to get cells from the cervix? Can't cells fall off from the endometrium? If a female is bleeding, menstruating, or any other heavy menopausal or postmenopausal bleeding, heavy menstrual bleeding, can she not have some cells falling down? So can you not get endometrial cells in your pap smear? You can, of course. Before that, what is this cell? Pink wala? Come on, you are now experts. Pink cell is going to be superficial cell. What is the blue cell? The blue cell must be the intermediate cell. And then can you see this is a cluster which is looking like a ball. I mean, many cells have gotten together and made a ball. This is a 3D ball-like cluster. This is a 3D ball-like cluster, and that is what we call as the endometrial cells. Endometrial cells are 3D ball-like clusters. Having said that, when do you maximally expect endometrial cells to come up? I would expect them during the first 5 days of the menstrual period. Most importantly, I would expect them from day 1 to day 5. That is the time when I expect them. However, that is the time when the bleeding is occurring. So when bleeding is occurring, I expect endometrial cells. If I do a pap smear in that time, I expect endometrial cells will come. But after the 5th day, when the bleeding stops, visible bleeding stops, you can't see bleeding, doesn't mean that the endometrium doesn't shed off. What are the remnants of the endometrium that are going to shed off? Remember, remnants of endometrial shedding continue even up till day 10. Whatever tiny cells have to shed off, that is going to occur between day 6 to day 10. So remember, in the first 5 days, you will see proper endometrium shedding off. You will see proper ball-like clusters. In the next 4-5 days, that is from day 6 to day 10, you are going to see something known as an exodus ball. And can you see how to identify this? The periphery is lighter and the center is darker. If you see this dual color ball, that is an exodus ball. That is remnants which are shedding off from day 6 to day 10. These are all the different cells. A very quick recap. If you see pink color cells, they are going to be the superficial cells. If you see blue color cells, they are going to be intermediate. If you see a cluster of round cells, those are parabasal. If you see a cell which can either look like tall columnar or it can look like honeycomb, that is endocervical. If you see a 3D ball-like cluster, it is going to be endometrial cell. That is on the first 5 days. After that, when remnants are being shed off, it is going to look like an exodus ball. That wraps up all your normal. Now, on the basis of this in gynecology, do you do something known as a maturation index? Yes, please remember, for that you will have to learn PIS. You know PIS, parabasal cell, intermediate cell, superficial cell. But that's it. That's what you do in gynae. So, what is maturation index? You are supposed to count 100 cells on a slide. So, for example, I'll give you some options and let's see if you can work it around. I counted a total of 100 cells. When I counted them, I realized that there was no parabasal. So whenever I'll write it, it will always be in this sequence: parabasal, intermediate, superficial. I realized there are no parabasal cells. I found 10 intermediate cells. Majority I have found as superficial cells. 100 I had to count, ho gaya 100. So which is the majority? Can you see there is a shift to the right because the majority that I'm seeing are the superficial cells. You know what are they an indication of? Superficial cells, the pink color cells, the pink for a girl, favorite color, that is a lot of estrogen. When there is an estrogen predominance, that is there, that is when I will suspect that, okay, more of superficial cells are seen. She's having more of estrogen right now. On the other hand, if I say that again, I count 100 cells, I have to go and write it in the PIS state. I said there are no parabasal cells. There are 90 intermediate cells and just 10 superficial cells. Where is the shift? I can see the shift has occurred to the mid-zone. The shift has occurred to the mid-zone. When you see intermediate cells shift to the mid-zone, this means this is a progesterone-rich state. This is a progesterone predominance that has happened. Maybe she is in, maybe pregnancy, if at all. Pregnancy is a progesterone-rich state. So that is what I analyze. Finally, if I see, again I'll write PIS. I see, say, let's say all 100 are parabasal and I did not see anything else. It's a total shift to left that has happened. Parabasal, all the estrogen progesterone cells are gone. See, neither am I seeing superficial, means no estrogen problem. Neither am I seeing intermediate, means no progesterone problem. I'm only and only seeing parabasal. That is going to tell me an atrophic smear. That is going to tell me atrophic smear. Basically, it's a smear that has been taken from a post-menopausal female. A smear that has been taken from a post-menopausal female. Let's do a recap. If you're seeing a shift to right, means lots and lots of superficial, lots of estrogen. If you're seeing a shift to the mid-zone, which is only intermediate, lot of progesterone. If you're seeing a shift to the left, means lots and lots of parabasal. Parabasal means no estrogen, progesterone. It is all gone, atrophic, post-menopausal smear. That is how you calculate the